CAR T cells, have just become FDA approved in 2017, initially for leukemia, now for various kinds of lymphoma. And they're evolving much more rapidly than I think other kinds of drugs. And we'll see much innovation in this area because of the fact that they're based on a patient's own T cells. Then people can make them better and better, but the primary medicine, if you will, remains the T cell that's injected. And so I think we'll see, you can make an analogy actually to computer systems where we initially had, for instance, Windows 95 and then Windows, et cetera. We're up to Windows 10. And so the cars right now, we're basically really car version 1.0. And every year I think we'll see new and improved and more potent and safer CAR T cells. At this meeting, there's a symposium today talking about a new one for instance, myeloma. It's expected that the next one that will get FDA approval will be next year for myeloma based on recent results published by a trial sponsored by Celgene. Another aspect of CAR cells is that they're, if you will, like an operating system, and you can just change what they're directed to, and the rest of the system remains unchanged. So virtually every kind of bone marrow or hematologic malignancy is being targeted in experimental trials around the world. Probably the biggest effort now is it's on myeloma, as I mentioned, but acute myelogenous leukemia, or AML, is being targeted at a number of centers, and I think we'll see FDA approvals and widespread use for all bone marrow malignancies in the next 5 years. So I think that there are two main areas where there are challenges. One being in solid tumors, where there's been some activity but no real home runs yet, if you will. And the reason for that is complex, but it involves the fact that the tumors are genetically more heterogeneous in solid tumors, and so CARs targeting multiple antigens are required. And so there are a number of approaches to solve that issue. So that scientific issue is being addressed worldwide in many research labs. The other issue actually is the manufacturing of CAR cells, and there's two main solutions there. One is they're being made more cheaply if they can be done by machines. Right now, CAR cells are made really by human labor. So you have to train technicians and scientists. And the only way they can really become much cheaper is if it's done in an automated way with robotics. So there's a lot of work to design those sort of instruments that would make these automatically. The other area, though, is using cells that aren't from the patient themselves, so-called off-the-shelf CAR T cells, which could be made from, for instance, cord blood or healthy donors. And then that would make the logistics much simpler.